Okay, can somebody confirm that you're hearing me?
Yep.
Great. My computer is working a little slow here because the lecture is still being made. Let's see, I think I got the syllabus. Oh, I got it right here, actually. So today what we're doing is Lab 01, Ubiquity of Microorganisms. Please remember that this Friday at, let's see, 8 a.m., you must turn in the worksheet for Lab 00, or you may be dropped from the class. For the Lab 01, it doesn't need to be turned in until Saturday night, I think 11:59 p.m. And normally the labs will be due Saturday, 11:59 p.m. of the week that we do the lab. So all the other labs will be due at that time. Lab 00 is an exception because I'm using that to determine who is attending class. Meaning I'm using it to get attendance. Any question about any of that? All right, let me see. I don't have it here. Really slow here. Let me close this because my computer is overworked. Go to Clark. I need to tell you to work or read the infectious disease project before next week. Next week, I'll be talking about the infectious disease project, and you need to read about the project and the instructions. And you can find that on Canvas, which I'm trying to get to, it's just on my computer's a little slow. And it's because it takes a lot of resources to convert the lecture file into a file that I can post on YouTube. Boy, I got a lot of mail, it looks like. I need to move this up. So you can find the infectious disease research project. I think it's under lab week 2. Oh, I just saw it. Where is it? There it is, right here. So click that link and take a look at this material. You don't have to print it out, but you should, at the very least, look it over. The infectious disease research project is a 100-point project, and it isn't due until November 5th, but you do need to look it over before I discuss it next week. Any questions about any of that? All right, if not, let's move to the lab. I think I'll get the worksheet open later. I'll look at the lab module at the moment. Where do I have two? Oh, I know I have two of those. Yeah, one's the meeting. Make sure I have the right file here. Yep. All right, lab module 1, the ubiquity of microorganisms. Like all of the lab modules, you'll have some reading material to read before reading the lab, meaning we assign some reading. However, the reading to this one is just a few pages. You need to read about culture media on page 157 to 162, and then how to obtain pure cultures from page 162 to 163. There will be video clips in this lab module. Your learning objectives: know what microorganisms are and that they are almost everywhere. Be able to describe the purpose of culture media. Be able to describe the terms colony abundance, colony diversity, and morphology. Be able to define the following terms: what is inoculation, ubiquity of microorganisms, something that's sterile, what is something that's sterile, what is isolation, incubation, in vitro, like in vitro growth, culture media. And when we say culture media, there are two different types, a liquid media and a solid media. The liquid media we call a broth. The solid media will have something solidifying the broth, and it's usually agar, but it could be something else. The culture media can be grown in a test tube or in a petri dish, a plate. If it's grown in a test tube, it could be grown as a slant or as a deep. And I think I'll talk about those later in this lab. Understand what a fastidious organism is, understand what non-selective media is, understand what ubiquitous means, and what's a colony. Any questions about any of these learning objectives?
Yes, two things. Can you share your screen?
Okay, can you get closer to your computer? I can barely hear you.
Oh, can you share your screen?
Oh, sorry, I thought I was.
And I have something else.
Go on and talking and I'm not sharing my screen, please let me know.
The tutorial pre-read, where is that at?
Like I was showing you where you could find a web page, and obviously you couldn't see it. So that didn't help you very much. So I meant to be showing you that. Log into Canvas, scroll down to week 2 lab, and then click on this link, Infectious Disease Research Project. Okay. So please let me know. That was really late. Somebody letting me know that you couldn't see the screen.
To answer her question about the pre-read section at the top of lab module 1, it's in your green book, your microbiology book from Clark.
Okay.
So when I go to
repeat that again, I couldn't hear you.
The pre-read section is what she had a question about. Those pages, the Tortora is your green book from Clark. That's your class book.
Correct.
Thank you.
All right. Microorganisms are organisms that are too small to be seen with the naked eye. This does include cellular organisms, viruses, and prions. We're also going to include helminths or worms in the classification of a microorganism. Okay, even though a worm is generally not microscopic, we'll talk about why we will talk about worms as a microorganism. And if you saw a window pop up, that meant the file for the lecture just finalized and I saved it down. All right, any questions about what we mean by a microorganism? All right, the phrase ubiquity of microorganism refers to the concept that microorganisms are almost everywhere. In this lab, we're going to investigate the abundance and diversity of microorganisms in common environments. We're also going to learn some basic lab protocols including the use of solid media, labeling lab cultures, procedures for disinfecting the lab benches, and procedures for washing our hands. You should know that a culture medium, also called a growth medium, is something you get in a microbiology lab that is sterile, a sterile laboratory preparation, so that microbes should not be growing in it, should not be in it, and it contains a variety of nutrients that are required for growing microorganisms. The culture media may be liquid, and we call these broths. A broth is commonly used in a test tube. We can add something to the broth to change it into a solid media, and usually we'll add agar to the liquid medium to make it solid. But you could use something other than agar. We'll talk about gelatin later in the lab, not today, but later. And another thing you could add would be starch. But generally, most solid media have agar added to make it a solid. We have a video you need to watch in this lab on preparing pour plates. And here we have some different ways of containing your media. These are Petri plates, and generally you'll have a solid media in microbiology in a Petri dish. That isn't always true, but generally it's solid in microbiology. You can also have a solid in a test tube, and this is a deep, and that's a slant. Both are solids. And then this is a broth. And a broth will almost always be in a test tube. We're going to talk about different media in microbiology. Today, we're only going to talk about general purpose media. A general purpose media is a media which is non-selective. So you can grow many things in it. It doesn't select for certain things to grow, and other things will not grow in it. The media is generally complex in composition, and it has things in it to support the growth of organisms, including peptone and yeast and beef extracts. And you can grow most non-fastidious microbes in a general purpose media. Now, a fastidious organism will not grow in a general purpose media. And we'll talk a little bit later about a fastidious organism. A general purpose media includes 3 types of media. NA, which is the abbreviation for nutrient agar with yeast extract added, or you could just have nutrient agar or NA media. It's the same, except it doesn't have a yeast extract added, or tryptic soy agar or TSA. These are all general purpose media. We use them a lot in microbiology because they are relatively cheap, cheaper than most other media, and many things can grow in them. Any question about that? All right. Whenever you're growing things in microbiology, you should use appropriate labeling. So it's vital that the Petri plate or the tube is correctly labeled. Always include your name or your initials, the date, the type of medium it is, like you could put Na for nutrient agar with yeast extract. The incubation temperature is optional, but you could put that. And you should put the inoculum, what you're trying to grow on the media. And here we have an example of that. The media is TSA. This is the temperature it's grown at, 37. There's the date. And this is what's growing on this plate, E. coli. And there's the name. You should either write on a petri dish around the edge of the plate, or on a test tube, you write on a piece of tape and then attach the tape to the test tube. Why you write on the perimeter of the base of the plate is if you were to just write all over here on the bottom of the plate, you'd have a hard time seeing the growth, and then the writing would interfere with the growth. Not interfere with the growth, would interfere with your ability to see the growth. So you write around the perimeter of the plate. You also write on the bottom of the petri dish. The reason is that the bacteria will be growing on the agar, which will always be in the bottom of the petri dish. And if the lid gets separated from the bottom of the petri dish, we want the writing to be with the bacteria. There once was an example where I was working in a lab and somebody was carrying a whole tray full of petri dishes, and I don't know, something like 100 or 200 petri dishes. And she was walking with them and then tripped, spilling the entire petri plates onto the floor, separating the lids from the bottom of the plates. And she had done all of her writing on the lids. So the experiment had to be thrown away because nobody knew what the experiment was anymore, which plate was which. So always write on the bottom of a petri dish. Any question about any of that? For inoculation, that's the placement of microbes into the sterile culture medium. That can be achieved in numerous ways. We'll show you a little bit about that. And then incubating it is just placing the dish in appropriate environment to incubate, such as an incubator, a place where you put the dish to incubate. I know that's using incubate a bit, but an incubator is a place where we put dishes to grow. I've got a picture here of an incubator. Let me scroll down. There's a fancy incubator. Fancy incubators can change the environment, like increase the CO2 in the air, decrease the oxygen once you shut the door. An incubator can also control the humidity. That's what this plate of liquid water is down there, to increase the humidity in the incubator. And this one you can see is a fancy incubator, which can control the temperature, which probably all incubators can do, as well as the humidity. But this one can control more things, like the amount of CO2 in the air. Most of our incubators in microbiology only control the temperature and the humidity. But fancy ones can control the oxygen and carbon dioxide. All right, so some important notes that during incubation, the agar plates will condense, water will come out of the agar and then will condense on the lid and then will drip down to the petri dish containing the bacteria. So then the bacteria will be spread around and not necessarily contaminated because the lid should be sterile, but it will definitely spoil the colonies that are growing up, mix the different colonies together. It could contaminate it, like if the water were to come to the edge of the lid and then drip down. So it increases the risk, but it doesn't necessarily contaminate the plate. So what we do is, when we incubate a plate, we will streak it out. This is a lousy plate, but hopefully you can see that. Streak it out, cover it, and then we turn it upside down and then place it in the incubator upside down. Now, if water comes out of the agar, it would condense on the top, which is where the agar is, and then the agar will wick the water throughout the agar so that there will be no drop forming. And even if one were to drop, it would drop on the lid where we have no bacteria growing, so it really doesn't matter. So that is why we grow plates upside down in the incubator, so that the water won't drop down on the plate and then mess up the bacteria growing on the plate. Any question about any of that? All right, so we mentioned a little bit about colonies growing on the plate. Colonies tend to have, bacterial colonies, I should say, tend to have rather smooth textured surfaces, although there can be exceptions. And generally, when you look at a colony, you can tell whether it is a bacterial colony. Let me blow this up. A bacteria colony where the edge is distinct and smooth, as opposed to a fungal colony where the edge is not distinct and the colony looks hairy or fuzzy. Another bacteria colony. Okay, these are not bacteria colonies. Sorry, I need to eat. These are fungal colonies, and this is a bacteria colony. So generally, when you can look, you can just determine whether the colony is bacterial or whether it is fungal. Any questions about any of that? So that's talking about colony characteristics. Let me state that a colony is made up of at least 1 million cells, and it could be 10 to 100 million cells making a colony. All of those cells in the colony may be descended from one or a few cells that were placed on the plate initially, and then it was allowed to grow into at least 1 million colonies before you can see it. Under 1 million cells, the colony will be so small, you really won't be able to see it. But after 1 million cells, it will be large enough to see. So right there, that's at least 1 million cells. And this colony is probably at least 10 million cells, maybe 100 million cells. All of them may have started from one cell or very few, and then the cells went through binary fission, splitting in two to make more cells. We have a little video for you to watch on binary fission. So watch that while you're doing the lab. And then take a look at the colonies. When we look at colonies, they will have colony characteristics. Each colony will have a shape. Is the colony circular, rhizoid, irregular, filamentous, or spindle shape? Each colony has a margin, that's the edge of the colony. If the margin is smooth, we call it an entire margin. If the margin is, I don't know, irregular, it's either undulate, lobate, curled, rhizoid, and filamentous. And if ever I'm asking you for the margin and it's rhizoid or filamentous, one or the other will be acceptable. I'm not going to ask you to try and separate these two from each other. And I'm not going to ask you to separate undulate, lobate, and curled from each other either. But you should be able to separate these from entire or these from rhizoid and filamentous. Elevation is the elevation of the colony. It could be flat, a raised mountain. It could be convex. It could be pulvinate, and that's just extreme convex, or it could be umbunate. And to see the colony elevation, you usually have to look at the colony from the side. Each colony has a size. It can be punctiform. Size of a punctiform is smaller than the tip of a pen. Small is about the size of the tip of a pen or a dull pencil. Moderate is larger than the size of a dull pencil or the size of a pen, but smaller than the eraser. And then large is the size of the eraser or larger. Each colony can have a texture. The texture is smooth or rough. Each colony can have an appearance, and the appearance is either glistening, meaning it's shiny, or dull. Each colony can be pigmented or non-pigmented. Pigmented means it has color, like purple or red or yellow. Non-pigmented is a colony that does not have color. That would be cream color, tan, off-white, or white. And then the colony can have optical properties. Most colonies are opaque. You can't see through it. Translucent, you can see through it, but it's like seeing through slightly muddy water. It's translucent. You can't see perfectly through it. And then transparent, that would be like looking through clear water. Very few colonies are transparent. So when we come and look here, this colony would be pigmented, it's yellow. This colony and that colony would be non-pigmented. The margin here would be entire. This one would be probably either filamentous or rhizoid. We can't tell the elevation really from this picture. That's probably pulvinate, is that what it's called? Pulvinate or convex, but it's hard to tell in a picture. The size, you'd have to look at this with comparison to a pencil, but you can see that one's punctiform. That one is probably small. Maybe that one's better for small. And then this would be very large. That one even is large, probably larger than a pencil eraser. Opaque. Let me say that one's opaque. I think most of these are opaque. Opaque, and this is shiny. That one's dull. Oh, let's come down here. I think I have a translucent one down here. Oh, there he is, right here, this big one here. This part here of the colony is translucent, and I don't know if that's the same colony or if that's a colony growing on top of this colony. This shows you an irregular colony right there. The margin is irregular. This would be entire, very regular. All right, any question about the colony characteristics?
I actually have a question. If we were supposed to identify like a black fungal colony on a lab or a test, would black be considered pigmented or not pigmented?
Sorry, I turned up my volume to try and hear you better.
Oh yeah, no problem. I can repeat myself if needed.
Black is not colored. I've never seen a black colony, but if you have a black colony, that would be non-pigmented because black is not colored.
Thank you so much.
Okay.
Just wanted to double check that.
I have another question. So if you go back to that Petri dish, the one you were just on, for the most part, were those colonies opaque besides that one that you pointed out was transparent?
I don't see any transparent colonies in any of here. None of them are transparent. Transparent are actually kind of hard to see, but I don't see any that are transparent. This one here, this part right here is definitely translucent. But this one, and that one, and that one, and that one are all opaque. So most of them are opaque. And in this one here, I think all of them are opaque. I could be wrong, but I'm not seeing any that are translucent.
Okay, thank you.
All right. Generally speaking, I would give you this table to answer this question unless it was a very easy question, like generally pigmented and non-pigmented. I might not give you the table, this chart to answer that one. The same with shiny and dull. Okay, but I don't expect you to remember the names of the margins, for example, so don't memorize it. I would give you the table if I were to ask you, I'd give you this chart if I were to ask you, what is the margin of this colony? Okay, so I'd give you this chart. Okay, and if ever I were to ask you for the size, I would have to put in a measurement here or put in a pencil because we don't really know how magnified this plate is. Actually, this plate looks very close to the real size of a plate, so I'm going to guess that these colonies are not magnified. So I'm trying to tell you, don't sweat that chart. I would only ask you something that's really easy on the chart. I'm not going to go through that. Let's talk about colony abundance. Abundance is simply the total number of colonies on the agar plate. So even if the two look different from each other, like this one looks different from that colony and that colony and that colony, but the abundance is just the total number of colonies on the plate. So let's come up here to this one. Now these colonies are probably all the same species, but that's probably a different species. And these is clearly a different species, and that's a different species. But the abundance would be the simple number of colonies on the plate. And I'm sorry, that's too many for me to count. I'm going to guess there's between 30 and 50 colonies on this plate. I went to that one because it's a little easier than this one. This one has a large number of colonies on the plate. I'm not going to attempt to try and count them here. But the abundance is just the total colony numbers. Like the abundance of cats in this picture here is what? The abundance of cats. Come on. 1,
it looks like
2,
7.
3, 4, 5, 6, 7. So the abundance is correct, only 7. The diversity is the number of different species. And in this picture, we have 2 different species, cheetah and lions. So the diversity is 2 cat species. Okay? So diversity would be the number of different kinds of colonies on the agar plate. And you look at the colony characteristics, and if the characteristics are really different, then it's a different species. This one's a little too hard to try and figure out the characteristics because there's so many. So let's come up here. But all of these yellow ones are probably the same species. All of these little white ones are probably a different species. So that's 2 in diversity. This one is probably a different species. In fact, I know it is, it's fungus. That real little one could be a different one than that one. So that's possibly 4. These 2 could be different. So that's 5. And then that's different from that one. So that's 6. So this would be, and that's different from that one. So that would be 7. So this looks like there might be 7 different species growing on this plate. Now, when I ask you a number, if you're close to 7, I will allow it. Okay? So if you say something like 6 or 8, that's good. However, if you say something like 3, I'm going to say 1, 2, 3, 4, 5. I'm going to say that's not enough. So that would be wrong if you say 3 or less. And then if you were to say something like 20, I'd say 20. How the heck do they get 20? 1, if that's different, which it probably isn't, it's 2, 3, 4, 5, and those 2 are probably the same. 6, 7, 8. That's probably the same as that one. So the most I can see is probably 8. Okay. So if you say something like 20, I'd say that's clearly wrong. So just make a good guess and I'll probably allow it. Any question there for diversity? Meaning if you're not perfect, it's okay. But if you're way off, I'll tell you, no, that's not close enough. Okay? And just use your reasonable guess based on the colony characteristics for different species. Like I said, this would be too different to figure out, so I'm not even going to try. But for the cats, it's easy. 2 species for the diversity. All right, know the terms. of this lab. And then complete the laboratory exercise. You don't need to look at the references. Oh, there's the answers to the clouds. Oh, and there's an answer to one of the plates, which I can go. Let's see. Let me get the questions now for lab exercise 1. And then when you're done going through the lab module, open up the laboratory worksheet. You can find the worksheet and the lab module in the lab week 1. There is lab module 1, and there's worksheet lab 1. Write your answers in the worksheet and then submit your worksheet in the link. I won't have the link because I'm an instructor. I'll have a grading page. So I've got download submissions here or the speed grader. You'll have over here, either on this side or it could be at the bottom here, you'll have a link for uploading your worksheet. Okay. Any questions about that? All right. And then answer the questions for lab module 1 in the worksheet. For procedure 1, we are asking the question, what's in the air you breathe? You do not have to answer this question, by the way. This is what we're trying to learn, but you don't have to answer that, all right? Answer the questions here where there's a question and then a space and generally a number. And normally it would be a number 1 and then A, but this is going to go under procedure 1. Question A, what colony abundance is on this plate? Below is the nutrient agar plate that was exposed to air and incubated for 24 to 48 hours at 30 degrees Celsius. And then we look at it and give me the colony abundance. As long as you're close, that's okay. But if you're far off, that's not okay. And then tell me what the colony diversity is on this plate. And it looks like that and that are the same colonies, probably that one. These two look similar, although they're different in size. Well, there's 1, 2, maybe 3. That's a different color, so 4. That's a different margin, so 5. This one's very different size, so 6. There's a different one, so that's 7. That could be different, so 7 or 8 is probably the diversity. And if you're far off from 7 or 8, I'll say make another estimate. Okay. All right, and then answer the question, what step would you take to reduce airborne contamination of a sterile plate that you are currently inoculating? Okay. So when you're inoculating this plate, what can you do to reduce contamination from the air while you're inoculating it? All right, procedure 2, what gets on your arms when you put your arms on the bench? You don't have to answer this question. This is what this procedure is going to try and answer. You label 2 NA plates, 1 bench before, 1 bench after, and then swab the bench with a cotton swab. You'll be then sterilizing the plate, and you swab the bench before you sterilize the workspace, and then you sterilize disinfect the workspace and swab it, and then swab the plate. And there we can see the before growth, meaning before we disinfect the workspace, and then after we disinfect the workspace. Answer the question, did table disinfection work? And then answer the question, what is the purpose for allowing the table disinfectant to air dry? I'm not sure the lab module directly tells you this, but if you read between the lines, you can sort of figure it out, or you could do go to the internet and ask this question on the internet, okay? But you should be able to figure it out. Procedure 3, what's on your hands? Label the plate before you wash your hands, and then after you wash your hands, and then touch the plates, and then look at the plates. Now, when you wash your hands, wash them appropriately, and you do need to have dry plates, dry hands, because I had some students who washed their hands and they didn't want to touch the paper towel because they're afraid bacteria could be on the paper towel. And then they touched the plate with wet hands, and the moisture on their hands actually wicked the bacteria onto the plates. And then they had more bacteria on the plate after they washed their hands. And that confused them. And I went through it and talked it over with them. And when they touched the plate, they were touching it with a dry finger or hand. And when they're washing the hands, they touched the plate with a wet hand. And the moisture does wick the bacteria onto the plate from your hand. And so you have to dry your hand and have the moisture on the hand similar to before you wash the hands, okay? And then answer the question, does hand washing work? You're not going to actually do this procedure, but we've got the pictures here for you to answer the questions. I guess I forgot to mention that here, before and after. So, does hand washing work? Okay, answer that question. And then suggest one reason why hand washing did not result in the complete removal of microbes. Meaning this hand washing is better than before you wash the hands, but it's definitely got some bacteria from your hands. And why might that be the case? If you obtain your answer following research, either online or in the book, please provide a brief citation of your information source. This can be answered just by thinking about it. Procedure 4, when you kiss your significant other, what exactly are you giving him or her? What's on your lips and what's on your tongue? So we have 2 plates here, which this one has been touched where the lips have touched the plate, and this one where the tongue has touched the plate. Please note, I've got a message here about this tongue plate, okay? If you don't read that note, you will get this question wrong. Which plate has higher colony abundance? Remember, abundance is simply total number of colonies, not the diversity. Why do you think there may be higher colony abundance on either the lips or the tongue? Meaning for whatever you answer for A. That is, what might cause the observed difference in colony abundance between the bacteria on your lips and on your tongue? And there's at least 3 reasons for why there's a difference in the number of bacteria on the tongue and the lips. And to answer this question, you must give me at least 2 of those reasons. And somebody's actually come up with a fourth reason, which to the life of me, I don't remember what it was, but there's a fourth reason for why there's a difference. But I can remember the three reasons. So give me at least two reasons for why there's a difference. And then why was the tongue plate incubated at a higher plate than the lip plate? So it tells you right here, the lips were grown, or the plate which was exposed to the lips was grown at 30 degrees C. And then the plate which was touched by the tongue was grown at 37 degrees C. Why do you think we grew them at different temperatures? And that's actually pretty simple when you think about it. Procedure 5, are there microorganisms in the soil? What you do is you get a little bit of soil, put it in some liquid, put a little bit of liquid on the plate, and then spread it around the plate. And that's what we've done here. Incubate it, and then count the colonies that grew up. And this is showing you the colonies that grew up. And then answer the question, what's the colony abundance? Is it even possible to accurately answer this question? Please explain your answer. And then B, is there greater abundance in the colonies from the soil compared to colonies from the lips? Answer that question and then submit your answers. Any questions about what you're doing? All right, if not, go ahead and work on the lab. I will be here until at least 8 o'clock. I think the lab goes to 8:20. And last time, everybody left the lab before 8 o'clock. Okay, I will be here until the last student leaves or until 8 o'clock. But after 8 o'clock, I will leave I will leave the Zoom session. Any question about any of that? All right, go ahead and get started.
Thank you.
And I'll be here to answer questions.